I can start.
Yeah, it's perfect. Okay. Good morning, good afternoon, good evening to everyone from wherever you're listening to. A warm welcome to all to the Progress and Prospects in Biology webinar series. Thank you once again for joining. We truly appreciate your support and kind cooperation to bring the webinar series to you. This is our 24th lecture of the webinar series. So those who are new to the webinar series, please subscribe to our channel and also listen to other archive talks that we have posted in this webinar series. During the talk, if you have any questions for the speaker, please post your questions using the chat box on YouTube, and we will moderate them towards the end of the session. With that, I would now request Professor N.R. Banerjee, the mentor and the convener of the webinar series, to give the welcome address and start today's session.
Thank you, Shaunak. And I must say that, every time these webinars are on, in the last moment, one of the devices will act up. So as we say, that it is always a good sign. When things are not working out in the last moment, then it forebodes well for whatever it is in the offing. So with that, a very warm welcome to you, Dr. Dasaradhi. I'm sorry, my Bengali pronunciation, I'm sure many of your Bengali colleagues will probably be also calling you Dasaradhi. So indulge me, please. And it is such a pleasure to host you today. And a very warm welcome to all our listeners from around the world. And like Shaunak says, progress and prospects in biology would not be what it is today without your active participation. So to all the scholars, to all the undergraduate and postgraduate students of biological sciences, to all the young and not so young and very senior academics who are listening in today, our speaker is going to talk about one of those creatures whose help scientists take to unravel the mysteries of life. So I think this is going to be, like one of those biology classes where one has to read up all the taxonomy and the morphological features and this and that, and then dig deeper and deeper and still deeper to get a glimpse of what we are all about. So we are looking forward to today's talk, and I hand over to Shaunak with a very warm welcome from myself and from the entire team of Progress and Prospects in Biology to our speaker today.
Thank you. Thank you very much.
Thank you, ma'am. It's my great pleasure to introduce today's speaker, Dr. Dasaradhi Palakodeti from the Institute for Stem Cell Science and Regenerative Medicine, NCBS, Bangalore. Dr. Palakodeti did his PhD with Dr. Pawan Malhotra and Dr. V.S. Chauhan at the International Center for Genetic Engineering and Biotechnology, New Delhi, where he studied the molecular analysis of cysteine proteases of Plasmodium falciparum and Burgay using RNA interference. He then moved to join Dr. Brenton Gravely's lab at the University of Connecticut Health Center as a postdoctoral fellow, where he got introduced to the Planarian regeneration model and studied the role of small RNAs and RNA binding proteins involved in stem cell function and regeneration. He's also one of the few who did the transcriptome sequencing of Planaria to annotate the Planarian genome at that time, which got refined later on in the literature. In 2010, he started his independent laboratory at InStem Bangalore, where he is currently an associate investigator, and his lab is focused on understanding the role of non-coding RNA and RNA-binding proteins in stem cell function and regeneration using Planaria and Hydra as a model organism. And also recently, they've been using embryonic stem cells to understand some of these functions. He received numerous awards, including the prestigious Wellcome Trust DVT India Alliance Intermediate Fellowship Award to start his lab, and also very recently the Tsrna Jayanti Fellowship in Biology from the Department of Science and Technology, Government of India in 2017. So with that, we are really looking forward to listen to Dr. Dasaradhi Palakodeti in the PPB webinar series today. And without further ado, over to you, Das.
Thank you very much. Thank you, Dr. Reena and Dr. Sonok, for this very kind introduction and for inviting me to this beautiful forum called Progress and Prospects in Biology. So I hope when I leave this talk, I will be adding my view of biology to this, and I hope you will enjoy what I'm going to add. So my lab in general is interested in understanding protein synthesis, and we think it's critical to regulate stem cell function and regeneration. In a minute, I will tell you why it is. So hence the title, Translational Regulators in Stem Cell Function and Regeneration, right? So, so why is protein synthesis important, right? When you take a stem cell and give it a signal, the stem cell undergoes these state transitions, and they subsequently differentiate into progenitors, which further differentiate into specific cell types. So when this differentiation, when the state transitions are happening, the first thing the stem cell has to do is bring down its protein repertoire. It could be proteins, structural proteins, or it could be an enzymatic proteins that are critical for maintaining stem cell in a stem state. One way of bringing the protein levels down is by degrading the protein. And while you are degrading the protein, there are transcripts that were already made, and the transcripts are being translated. So it becomes counterproductive to make the protein while you are degrading it. Hence, translational regulation becomes a key factor here in order to regulate the protein levels, right? So that's what my lab is interested in understanding, how this translational regulation happens, which is critical to drive stem cells from stem state to a differentiated state. In order to address that, we use different model systems. So one of the model systems, as Dr. Reena and Dr. Sonok said, which my lab extensively uses, is Planaria model. I'll tell you subsequently why we use Planaria as a model. And another model system that we recently started using is embryonic stem cells, especially the mouse embryonic stem cells, right? Because they are pluripotent stem cells and they can differentiate into any cell types. So translational regulation, this is the most simplistic form of translation regulation that you see on my computer screen here. And it can be broken down into three pieces, and basically the three places where the regulation can happen. One is at initiation state, where ribosomes are loaded, and the elongation state, where the ribosomes actually run through the transcript. And the other is a termination state, where the ribosomes are opened up, and then they are reassembled at the initiation state, right? And these are the three layers at which the translation can be regulated. And if you just take this simplistic model and blow it off, you can see the complexity of regulation here. So you have a specific set of proteins which are critical for assembling the 40S ribosomes. And the 40S ribosomes gets assembled in a cap-dependent manner. And the 40S ribosomes runs through the mRNA, stops at the AUG site, right? And that's where you add your 60S and the elongation starts. Elongation goes through, stops at stop codon and gets dissociated. So there are secondary structures which are within the mRNA, which are called as either G quadruplex structures or iris kind of elements that potentially regulate how the translation is happening and how proteins are loaded. In addition to that, you also regulate the initiation factors, and by regulating the initiation factors, you can regulate how the ribosomes are loaded and hence regulate the translation process. And a great deal of regulation also happens at the 3' UTR regions, again in the form of G quadruplex structures, or also you have a lot of other non-coding RNAs such as microRNAs and other long non-coding RNAs that can interact with the 3' UTR regions and block how the translation process is happening. So my lab previously have studied in RNA binding proteins, and some of the work has been published. One of the RNA binding proteins where my lab has extensively looked at is this poly A binding proteins that you see here, which binds to the poly A tail and regulates how the translation is initiated. So we have shown that there are specific poly A binding proteins that are expressed in the stem cells that are essential for epidermal progenitor formation and also essential for the epidermal integrity. In addition to that,
Excuse me.
hello?
Sorry to interrupt you. I think the slides are not visible, the transition.
Oh, the slide, you can't see the slides?
No, I think it's only the first slide. Yeah, now it is coming. Translational
Okay.
regulation. Maybe
So,
it's full screen somehow.
it's not working. So it's gone now? Can you see?
No, we can see the slide, but it's very small. Like we can see all the slides on the side.
Okay, so I don't know what I'm doing. Oh. How about now? Can you see now?
Yeah, we can see it, but it's just small. So is there an option of like full screen? That keynote live or play, I don't know what that two works.
I'm trying. Okay. No, you can't still see?
No, it's the same.
No.
Or maybe we can zoom it from there. It's 51%. Maybe make it 100. That may work.
Okay. So you can. So you're seeing a smaller screen. That's what
Smaller
you're saying.
screen, yeah. So you can see the translation regulation, that slide.
Okay. So let me see. Still not able to see, right?
No.
But yesterday we have gone through this, right?
Yeah,
Let
yeah.
me 100%.
Yeah, now it's fine. Perfect.
Now it's fine?
Yeah, perfect. Yeah. Sorry
You
for
can-
that. Yeah,
no,
I can
no.
see. Yeah.
Yeah.
I can see.
Okay. Yeah. So please stop me if you have any issues of that sort. Okay. Yeah. So the other aspect that my lab is also looking at is the length of poly-A tails. And Vairavan, a PhD student from my lab, has done this beautiful work where he has annotated the 3' UTRs, and he's now looking at poly-A polymerases that actually regulate the length of a poly-A tail. And he has shown beautifully how regulating the length of a poly-A tail could influence how the neoblasts are undergoing proliferation and differentiation. And today, what I'm going to talk about, my lab also does extensive work on microRNAs and how they regulate organogenesis. And we have identified this MIR124 family of microRNAs that regulate how the brain formation happens in Planaria. So today, what I'll be talking about is this very interesting class of small RNAs, which are actually derived from tRNAs. So we all know that the tRNAs carry amino acids. So what we have found in last, say, couple of years is that these tRNAs can be broken down into multiple pieces, and each piece can go back and regulate the translation. And we have shown that this is expressed in the stem cell populations of both mammalian stem cells as well as planarian stem cells, and actually regulate the process of stem cell differentiation. So today my talk in next, say, 35 minutes or 40 minutes will be mostly towards understanding how the tRNA-derived small RNAs regulate translation and how does that regulate stem cell function and regeneration. So the focus of the talk, I will be talking about how we identified and studied the function of this tRNA-derived small RNAs, which are also called as tsRNAs, in mouse embryonic stem cells. And I will touch a bit, this is still work in progress, I will touch a bit about how the biogenesis of these tsRNAs happen. How does the breakdown of tRNA happen to make tsRNAs, and what is the hypothesis we have behind that? And then I will talk about the function and biogenesis of these tsRNAs in planarians, right? Which also shows how evolutionary conserved this small RNA populations are. So what are tsRNAs?
I think
So
sorry, it's not transiting.
It's not transiting.
No, it's just getting stuck. So I think we have to click individual slides, I guess. Yeah, now we can see the tsRNA slide.
You can see tsRNA slide. Okay. Is it full screen or
Yeah,
it's a
it's full
small
screen. Yeah. So I think we have to click in individual slides to go.
So if I blow it off, you can't see, right? That's what the
No.
problem.
Yeah.
Now, can you see the TSRNA slide now?
Yes. Yes, I can see that.
Okay. So the transitions are not happening. That's what you're
Yeah.
saying.
Yeah.
So can you see my next slide? Can you
No,
see
it's
the?
still the TSRNA.
Oops. Okay. Okay. I mean, let me see. If
Yeah.
you can see this way, maybe I will just show it here. Okay. Can I play from here? No. Okay. So let me see what I can do.
Maybe I think we can click individual and then that will work.
Okay, you can see that, right?
Yes, I
So
can see the TSRNA slides.
yeah, TSRNAs, right? Okay, so so the next part is so what are these TSRNAs and what is their role, right? I mean, it has been shown that the tRNAs can be broken down into multiple pieces. It can be broken down into this 18 to 22 nucleotide species, which are mostly derived from the 5' and the 3' arm, which you see here. And there are also these 25 to 34 mer species that are derived from the 5' arm, and this actually break right in front of the anticodon arm. This has been shown, and most of my talk will be about this 25 to 34 nucleotide species. And this species has been shown to play a role under stress regulation. So if you take a cell and put it under any kind of a stress, either it's a DNA damage or a hypoxia conditions, it has been shown that the species go up and thus shut down the translation, right? But what we have interestingly found, so can you see the transition now?
Yes, yes, it's perfect.
So now can you see the
Yeah,
transition?
perfect. Yeah, yeah.
Yeah. So what we have seen is this: when you take an embryonic stem cell and treat them with retinoic acid, the embryonic stem cells undergoes differentiation into neural progenitors. So within 48 hours of treatment of embryonic stem cells with retinoic acid, we see a surge in the population of this 32 to 33 mers going up, right? And they go up as the cells undergo differentiation. That means they go up high as the ESCs differentiate within 48 hours towards the neural progenitors. So first, and then we ask the question, what is this 32 mer population about? Initially, we thought they're piRNAs, but when we map them back, we realized that they are actually coming from the tRNA population. The first thing that came to us is it could be a degradation product. Just to make sure that it is not a degradation product, we took it and mapped it to the tRNA population, and we saw that most of these tsRNAs actually come from the 5' arm of the tRNA, and they end just before the anticodon arm. So this tells us that there should be a function that must be associated with it, because if there are degradation products, they should fall all over the place. But that's not what we saw. And we also validated this both by qPCRs as well as by northern hybridizations, right? So what is the function associated with these tsRNAs? Do they have any functions in terms of stem cell activity? So we know that, and in order to establish that, what we have done is we took ESCs, we treated the embryonic stem cells with the antisense oligos against the tsRNAs, and we have treated them with retinoic acid. If tsRNAs are essential for ESC differentiation, you should see when you treat with retinoic acid, they should differentiate, right? If you put an antisense oligo, you should block the differentiation of ESCs, right? So how do you measure the embryonic stem cell activity? We measure by a process using alkaline phosphatase activity. Basically, what happens is in an embryonic stem cell state, you have a higher levels of alkaline phosphatase activity. As the stem cells undergo differentiation, the alkaline phosphatase activity go down. But now, if you treat the ESCs with the antisense oligos, you should retain the stemness, so you should see an increase in alkaline phosphatase activity even when you treat with the retinoic acid. And that is what we have seen here. We have seen that when you actually treat ESCs with antisense oligos and put them in retinoic acid, they slow down the process of differentiation, which is actually measured by an increased levels of alkaline phosphatase activity. We also did the converse experiment. What we did is, instead of putting an antisense oligo, we have put a mimic. And instead of putting them on retinoic acid, we have put them on a leaf, which maintain ESCs as ESCs, right? So if you put a mimic, so basically you are ramping up the concentration of tsRNA. When we did that, even though the ESCs are there in leaf, they started undergoing differentiation, which is measured by a decreased level of alkaline phosphatase activity. What these two experiments, one is an antisense experiment, another is a mimic experiment, is telling us is that there is a functionality associated with these tsRNAs in controlling how the embryonic stem cells are actually differentiating. So in summary, we have identified this new class of small RNAs, which are 30 to 34 nucleotide in species. We have seen that they are enriched as the ESCs undergo differentiation. And we have also shown that they are very strongly associated with polysomes. I have not shown that data here. And we also have shown that they suppress overall translation. And by using antisense oligos and mimics, we have shown that they are critical for the ESC differentiation. Now, the biggest question that remains is, how does tsRNA regulate the differentiation of ESCs? In other words, what is the interactome of this tsRNA? Because by understanding the interactome, you can also understand the functions associated with it. So in order to understand the function, so you can see the slides, right?
Yes,
Tsrna, you can see the slides?
Yeah, we can see it.
Yeah, thanks. In order to understand the function of the tsRNAs and in order to understand what its interactome is, so we have done a huge array of high throughput studies here. So what we have done is we took ESCs, and we took ESCs that were treated with retinoic acid after 48 hours. We made cell extracts. We took a biotinylated tsRNA and we incubated with this extract and pulled down with streptavidin, and we have done transcriptome sequencing to ask the question, is this tsRNA anyways interacting with the mRNA? And we have identified a completely different pools of mRNAs that are interacting with tsRNA both in the ESC condition as well as an RA condition. We are finding two different pools of mRNA that are interacting with tsRNAs in the ESC as well as in a retinoic acid treated condition. So when we went back and asked, what are these mRNAs that are associated with tsRNA in ESC condition, we found that those mRNAs that are associated with tsRNA in a ESC condition are the ones that are essential for differentiation, suggesting potentially that these tsRNAs might be interacting with differentiation specific mRNAs and blocking their translation so that the ESC is not differentiating, right? And now we ask the question, what is this tsRNAs doing in a differentiating condition, which is when you treat with RA? What we interestingly found is that tsRNAs started interacting with the mRNA that are associated with pluripotency factors. So you need to block the translation of these pluripotency factors in order to push the cells towards the differentiation state. In addition to the pluripotency factors, we also found some of the differentiation responsive genes that are associated with the tsRNA. If you go back and ask what are these differentiation specific genes, these are the genes that are essential for the mesodermal fate and the endodermal fate. So what we are doing in this experiment is we took ESCs and treated them with retinoic acid. So basically we are pushing the cells towards the ectodermal fate. So what interestingly we found is when you take the tsRNAs at 48 hours treatment of retinoic acid, we found that these guys are actually blocking pluripotency factors, and they're also blocking the other linear specific factors, suggesting that these tsRNAs that are upregulated has specific functions in terms of driving ESCs towards differentiation to a specific state here. So now the question is, how does it interact with mRNA? So the two ways it can interact with mRNA is they have a complementarity with the mRNA. The other way it can interact with mRNA is either through a RNA binding protein. to rule out or to see whether the first one is true, that is, it is binding with some complementarity, we went and used bioinformatic tools to understand whether they have a complementarity or not. And we haven't found any complementarity that can justify that this is the way it is regulating the mRNA. So the other possibility is that it is interacting with a set of RNA binding proteins that could be actually modulating the translational regulation function. So in order to identify that, what we have done is we took tsRNAs, we did the same experiment, but now instead of pulling down the transcript, we actually took the lysate that has pulled down with streptavidin, and we have given it for proteome analysis. And we found that both in ESC condition as well as in a RA condition, these tsRNAs are interacting with different sets of proteins here, right? One of the top hits where the tsRNA is actually interacting with a RNA binding protein in a retinoic acid treated condition after 48 hours is the specific protein called IGF-2BP1. So what is this IGF-2BP1 and what is the role of this IGF-2BP1 and why is it interacting with tsRNA is something I'm going to talk about. So what is IGF-2BP1? It's called insulin-like growth factor 2 mRNA binding protein. In a nutshell, it's an RNA binding protein, and it has been shown to be highly upregulated in cancer cells. And if you block IGF-2BP1, it actually blocks the proliferation of the cancer stem cells or cancer cells. So how does it do? We know that IGF-2BP1 binds to c-Myc mRNA, and c-Myc is one of the cell cycle regulators and critical for cell cycle, right? The IGF-2BP1, along with other RNA binding proteins, bind to c-Myc mRNA, stabilizes the c-Myc mRNA, and loads the c-Myc mRNA into the translating complex, and does allow protein synthesis to happen. So now it's a very interesting hypothesis that we have in place here, right? That is, in a ESC lift condition, you have IGF-2BP1, which binds to c-Myc mRNA and allows translation to happen. What happens in RA treated condition? You have tsRNAs that are upregulated. We know these tsRNAs interact with IGF-2BP1. So we hypothesized here that if this interaction happens, it destabilizes c-Myc mRNA. As a result, the c-Myc mRNA doesn't undergo translation. So if this is the hypothesis, how do we prove that? We can prove by doing 3 different experiments here. One is, does IGF-2BP1 selectively interact with tsRNAs in RA condition? Is one. Does IGF-2BP1 interact with c-Myc mRNA differently in a RA condition? Is a second experiment. The third experiment is, if this is true, the c-Myc mRNA should be dislodged from the translating complex in a RA-treated condition. So that's what we have checked in the third experiment. So let's see how these 3 experiments panned out. So in order to understand whether IGF-2BP1 interacts with tsRNA and c-Myc, so we have again made lysates from ESCs and retinoic acid. We have pulled down IGF-2BP1. In one case, we have isolated small RNAs, and we have done small RNA sequencing. In other case, we isolated the transcripts and we did qPCRs with the c-Myc mRNA. So what we found is the IGF-2BP1 heavily interacts with tsRNA in a RA-treated condition. And very interestingly, the IGF-2BP1 interaction with the c-Myc mRNA here goes down as you treat the cells with retinoic acid, suggesting that in a retinoic acid treated condition, the c-Myc mRNA is loosely interacting with IGF-2BP1, and IGF-2BP1 is interacting a lot with the tsRNAs. That proves the first 2 experiments, right? The second experiment, the third experiment that we wanted to do is, what is happening to c-Myc mRNA in a translating complex in both ESC as well as retinoic acid treated condition. So what we did is, we took ESCs, we took retinoic acid, we did polysome profiling, we isolated RNP complex, which is a non-translating complex, we took ATS complex and we took polysome complex, and we asked, where is the c-Myc mRNA in these 3 complexes, right? So So whether it's moving from non-translating complex to a translating complex in retinoic acid treated condition versus ESC, right? What we found is when you treat with retinoic acid, the c-MYC mRNA presence in a non-translating complex is going up, and in a translating complex, it's actually going down. So what are we proving here? We have proved that the IGF-2BP1 could potentially interact with tsRNAs, and their interaction actually destabilizes c-MYC mRNA, and the c-MYC mRNA moves from a translating complex to a non-translating complex in a retinoic acid treated condition. As a result, you see a decrease in the protein synthesis of c-MYC mRNA. So that's what we have shown here. But this is all very correlative, right? If this model is true, now if you treat the cells with antisense oligo, you should dislodge the tsRNA from here. And if you dislodge the tsRNA from here, the c-MYC mRNA should go back into the translating complex, right? So that's the model. So in order to prove that, we took ESCs. In one case, we treated with retinoic acid. We did polysome profiling. In other case, we treated the ESCs with antisense oligos, and we treated them with retinoic acid, and we did polysome profiling. So what happens when you treat with antisense oligos? The c-MYC mRNA should go back into the polysome complex. And when we did the ratio of non-treated versus treated with antisense oligos, what we found is in an RA condition, in an antisense oligo treated condition, you see that the c-MYC mRNA is back into the polysome complex from the non-translating complex. So this experiment actually tells us clearly that the tsRNAs regulate the translation of c-MYC mRNA by its interaction with IGF-2BP1, and its interaction with IGF-2BP1 destabilizes c-MYC mRNA. As a result, the protein synthesis doesn't happen. So this is just a tip at the iceberg in terms of understanding tsRNA function. We also now interested in understanding how linear specification happens here, right? So what have I said so far? What I have said is when you take embryonic stem cells and you treat with retinoic acid, right? What we see here is the translation of stemness genes goes down. And at the same time, you also see the translation of differentiation markers go up. And what we see is that the tsRNAs that go up here is actually regulating the translation of a stemness gene, which is critical for pushing the stem cells towards the differentiation state. So that is where we have in terms of understanding the tsRNA function. But one of the bigger questions that remains unaddressed here is how is this biogenesis happening, right? So in next couple of minutes, I will put our hypothesis across in terms of understanding how the biogenesis of tsRNA happening here. So that brings us to the second part of my talk, which is understanding how the tsRNA biogenesis is happening. So if there is an animation, this would have been better, but let me try explaining you this. What is the enzyme that cuts tRNA to make tsRNA? And it has been reported that there is an RNA binding protein, which is an endoribonuclease called angiogenin, which is essential for angiogenesis, was shown to be essential for cutting the tRNA into tsRNA, right? But when angiogenin cuts the tRNA into tsRNA, it leaves this 2'3' cyclic phosphate group. And this species cannot be sequenced directly. In order to sequence this species, you need to treat the small RNAs with T4 polynucleotide kinase, and then you can sequence. So when you do this protocol, you are actually identifying small RNAs that are derived from angiogenin. And we also think that the tRNAs are also processed by another endoribonuclease other than angiogenin, and that will not add 2'3' cyclic phosphate, and that can be sequenced by a conventional sequencing method. So what we have identified, I'm just summarizing the result here. What we have identified is when the stem cells undergo differentiation, the pool that actually goes up here is an angiogenin independent tsRNA pool. It's not angiogenin dependent pool. In fact, what we have seen is the angiogenin-dependent pool goes down and comes back, right? So now you have this interesting condition here. You have a tRNA that can be processed by an angiogenin to make tsRNA, right? And you also have a tRNA that can be processed by another endoribonuclease to make tsRNA. And these two tsRNAs are coming from different pools of tRNAs, and they actually regulate the process of stem cell function. So one of the big questions that remains to be addressed is, how is the regulation of tsRNA biogenesis happening? What decides what enzyme cuts it? What decides how much of tRNA has to be processed into tsRNA? Are some of the burning questions my lab is interested to address, right? So do we know anything about this, right? So what has been shown here is this: the tRNA is the heavily modified RNA molecule inside a cell. And it has been shown previous' from Blancos et al.'s work is that when you have a tRNA, when the tRNA is modified here at a 5' methylation at a cytosine just before the anticodon arm, and this modification happens by a methyltransferase called NSEN2. The angiogenin, which is an endoribonuclease, cannot recognize the tRNA. As a result, it doesn't cut, right? So other possibility is that when you don't have a modification, the angiogenin can actually cut and generate a tsRNA. So how do you regulate now? You regulate by changing the modifications on the tRNA. So now the question remains is, what are those modifications? And we know that the tRNAs can be methylated, they can be thionylated, they undergo pseudouridylation and many other things, right? So when we went back and asked the question, is there any change in the levels of enzymes that are essential for thiolation, both in the ESC condition, in a retinoic acid treated condition? And what we found is the enzyme levels are not changing at all, at the transcript levels at least. And we also asked if there are any methylases and demethylases whose levels are changing. And what we found is both methylases and demethylase levels are also not changing, basically suggesting that the enzymes that are critical for these modifications are not going any changes in their levels. Then what is changing here? So that's where we got interested in understanding how this enzyme kinetics work here. Both demethylases and methylases need cofactors. For instance, methyltransferases requires this cofactor called SAM, right? And this is a methyl donor. And if you have a limited number of SAM, you can actually block the methyltransferase function. The other way to regulate demethyltransferases is by regulating alpha-ketoglutarate, which is a cofactor for demethyltransferases. So now you don't really have to change the enzyme levels inside a cell. You can actually change the enzyme activities by actually changing the levels of methyltransferase cofactors such as SAM and alpha-ketoglutarate. And there is this interesting work that came up recently where they have shown is when you take an ESC and deplete the ESC out of methionine, the embryonic stem cells undergo differentiation, and they said that this is happening potentially because you are changing the status of your methylations and demethylations in the nucleus. As a result of changing the epigenetic status, you are actually promoting the ESCs to undergo differentiation. And recently from INSTEM, Dr. Sunil Lakshman has shown that the tRNA modifications can also act as a sensors for changing amino acids. And as a result, he has shown in the yeast system that the tRNAs can be cleaved, right? Using this two work, we wanted to suggest here this, that when you actually deplete methionine, in addition to the epigenetic changes you are seeing, you are also seeing the changes in the modifications of the tRNA. And this change in the modifications in the tRNA are actually leading to the differential breakdown of the tRNA into tsRNA. So how do we test that? So first we took the embryonic stem cells, we treated with different concentrations of methionins. As we deplete the methionine levels, we could actually see at 0.025 millimolar methionine in the culture medium, you see that the embryonic stem cells start undergoing differentiation. And we have also shown by alkaline phosphatase activity and colony size that indeed the cells are actually undergoing the differentiation. So now we know that when you deplete or bring down the levels of methionine to 0.025 millimolar, you allow the ESCs to undergo differentiation. So what we have done is we took ESCs that were depleted with methionine, and we did small RNA sequencing and asked the question, are there any tsRNAs that are differentially enriched? And I'm just summarizing the result here. We found that there are tsRNAs that are upregulated when you deplete, and there are tsRNAs that are downregulated when you deplete, and there are certain tsRNAs that don't change between the control and the methionine depleted cell cultures here. So what is interesting is this. I am saying that when the work has shown that if you add a methyl group to the tRNA, you prevent the angiogenin to break it up, right? So I understand upregulation. What is interesting here is how is the downregulation happening? So we took the tRNAs, we took the tsRNAs that were upregulated and downregulated, and we asked, is there any base residue preferences? And we have actually seen that there are differences in the base residue preferences between the tsRNAs that were upregulated and the tsRNAs that were downregulated. So what is next, right? So we also went back and asked the question, what are these tsRNAs? And I'm going to bring two different examples here. One is tsRNA arginine. This is a tRNA that carries arginine amino acid that is broken down into tsRNA here. And very interestingly, there are two isoforms. One is arginine TCG is actually upregulated. This tsRNA is upregulated, and you can see this ARG CCG is actually downregulated. The isoforms, the two isoforms, the tRNA isoforms that actually carry arginine, in one case they are downregulated, in another case they are upregulated. So when we went back and asked, what do we know about the modifications? What we found is that in one case where it is upregulated, you have a cytosine that can potentially be methylated, right? In other case where it is downregulated, the same position has uridine, and that can be thylated, and there is a methylation event that can happen at the D loop of this. So here, based on where you actually methylate the tRNA, where you add a thiol group on the tRNA determines whether the tRNA is actually upregulated or downregulated. It's not that always methylation means preventing the cleavage. Methylation can also enhance the cleavage, and that's what we are trying to bring out here, right? And whether it enhances the cleavage or whether it actually brings down the cleavage depends on the position where you are actually making the modifications. And we are doing mass spec on the tRNAs to understand where these modifications are happening, so that we can go back and understand how this regulation is happening here. So just to summarize quickly, and this is our working hypothesis, you take pluripotent ESCs and you deprive it of methionine, and you see that ESCs are undergoing differentiation. And what we have found is, we have found two sets of tRNAs that are being derived into small RNAs, and one set is actually going up and other set is actually going down when you deprive methionine. And this up and down is being decided based on where the methylation is happening on the tRNAs. So understanding where these methylation events are happening or where these modifications are happening can shed actually a light in understanding how the biogenesis of tRNA happening inside a cell, right? So this is all about the aspect of understanding the biogenesis of tRNA, and this is still in work in progress, and we are trying to understand what regulates the modifications and what is the interactome that actually tells where the modifications have to happen. So coming back to the last part of my talk, which I will try to wrap up in another 5-10 minutes, is what do we know about the functional conservation of these tsRNAs? So the simple question is, does this tsRNAs regulate stem cell functions across metazoans, or is this specific property that you actually see in mammalian stem cells? So the big question is, are they functionally conserved? Since my lab extensively uses Planaria as a model system to understand the aspects of stem cell function and regeneration, we decided that we wanted to go back and look at what tsRNAs does in the planarian populations, right? So what is Planaria? Planaria is a flatworm, right? And the species that we use in our lab is called Chimidia mediterranea. And why we picked up this animal is because this animal undergoes extensive regeneration. As I have shown in the slide here, you can take Planaria and you can cut the Planaria into tiny pieces, and each piece here can regenerate back into a complete animal, right? And it has been shown by many labs across the globe that these animals carry this specific cells called as neoblast, which are pluripotent in nature, and these cells are critical for the regenerative property of the animal. So in a nutshell, if you take Planaria, you cut Planaria into 3 pieces, what has been shown is that the pluripotent neoblasts, which are red in color, undergoes this differentiation into committed neoblast. And the committed neoblast further differentiates into early and late progenitors and does actually form the missing tissue and organs. So by using single cell transcriptome, which actually came from 3 different labs, what I'm showing here is only the data from 1 lab. Why I'm showing this is to show the complexity of the stem cells here. So what this slide shows here is that, which came from Alejandro's lab, is that you have a clonogenic neoblast, which is a pluripotent neoblast, which can divide or differentiate into committed neoblasts here. And each committed neoblast can further give rise to these progenitors and also give rise to the specific cell types, right? So you can now sit and imagine what cell state transitions and differentiation, how critical it could be for Planaria, and all these translational regulators should and must play an important role in the progression of the stem cells into a differentiated populations, right? So what am I going to address here? The first question I wanted to address here, are tsRNAs present in Planaria? The next question is, if they are present in Planaria, are they expressed in stem cells? If they are expressed in stem cell, how is their biogenesis regulated, right? So in order to address this, we took 2 strategies here. As we know, these animals can undergo extensive regeneration. So we took an uncut animal and we sliced them into small pieces. And this has been done in collaboration with Dr. Jochen Rink from Max Planck Dresden. Now he moved to Göttingen as a director. So why we did this experiment is because we wanted to know if there are tsRNAs, how spatially they are distributed, right? So what we have done is we have taken the Planaria, we cut into 11 segments, and we took each segment and we did small RNA sequencing to identify, are there any tsRNAs distributed across the Planaria? And we also want to look at the presence of tsRNA in the regeneration conditions. So what we have done is we cut the Planaria, we just took the blastema, which is a regenerating part of the Planaria, and we have done small RNA sequencing to identify whether there are any tsRNAs during the process of regeneration, right? But there is a problem in the field, is that when we started looking for tsRNAs in Planaria, we realized that the tRNAs were not annotated properly in the Planarian genome. So the first thing we have done in order to overcome that problem is we went ahead and we have annotated the tRNAs in the Planaria genome, and we found that there are around 457 tRNA loci in Planaria. And what you see, the histogram here, is the number of tRNAs for each amino acid that they actually carry with them, right? So we also build this interesting codon table here, and we have validated some of the tRNAs to show that these are indeed actually the tRNA populations. So now what we have tRNAs with us. We have a tRNA library, or we know all the tRNA loci from our bioinformatic analysis. So now what we did is we took the small RNA populations that were generated from the salami section here, and we have mapped it back to the tRNA loci that we predicted, and we asked, are there any small RNAs that came from the tRNA? And we found that there are indeed around 3 to 5% of small RNAs that are actually derived from the tRNA loci, and we can confidently call them as tsRNAs. And very interestingly, we also found is this: in addition to 25 to 35 mer population that we found, we also found 18 to 24 nucleotide population, which are the smaller populations that I have shown at the start of my slide, right? So what did we find here? We found that the tRNA can be broken down into 3 pieces in Planaria. One piece is 25 to 30 nucleotide species that is ubiquitously distributed all across the fragments of the Planaria, right? And they end right before the anticodon arm. The other is 18 to 24 nucleotide species, and this is an interesting species. One part of 18 to 24 nucleotide species that you see here are derived from the 5' arm. The other part, we call it as intermediate tRNA species or fragments. They are derived right after the codon and stop right before the T-size loop that you see here. So these are called as intermediary tsRNAs, which are 18 to 24, and we call them as 5' TERs, which are 18 to 24 here. And this is our population, which we initially characterized, which is 25 to 30 nucleotides, that comes from here and all the way goes down till the anticodon arm. So what we interestingly found when we did small RNA sequence and mapped it to tRNA loci is that we found 3 different pools of tsRNAs, right? And the question that we asked is, are they all coming from the same tRNA? Are they coming from different tRNAs? And what we interestingly found is this: the 5' tsRNAs, which is 25 to 30 nucleotide species, and 18 to 24 nucleotide species are actually coming from different pools of tRNA loci. That means there are tRNA loci that are being differentially processed. One loci is making 25 to 35 nucleotide species, and other loci is actually making 18 to 24 nucleotide species. So we also took all the small RNAs and we looked for the base preferences at the 5' end. Why is looking for base preference for 5' end important? Because by seeing what is the first base, you can determine what could be the enzyme that is cutting it. For instance, when we looked at the first base of the small RNA populations, what we found is the ITAUs, which are 18 to 24 nucleotide species here, have a very strong enrichment for U at the 5' end. This is an 18 to 24 nucleotide species, and it has a strong enrichment for U at the 5' end, which suggests that this species could potentially be formed from the dicer and organate product. So what is summarizing so far? We identified 3 different pools of tsRNAs in Planaria, right? One is a 25 to 35 nucleotide species, and other is an 18 to 24 nucleotide species. In the 18 to 25 nucleotide species, we have something that is coming from the 5' arm, and we have something that is coming from the intermediates of the tRNA species here. And we found that these ITAUs have a strong enrichment for U, suggesting that these are coming from the enzymes, possibly either by the cleavage of dicer or by the cleavage of organate, which we need to figure it out, right? So are they differentially expressed during regeneration is the next question that we have. So what we did is we cut the Planaria, we took the blastema, and we took the blastema, that is a regenerating tissue, at 3-hour, 12-hour, 24, 2-day, 3-day, 5-day, and 7-day. And we have done small RNA sequencing to identify the tsRNA populations that are enriched during the process of regeneration. And to our and we again, during the regeneration, we found both the species of tsRNA. One is 25 to 35 species, and other is 18 to 24 nucleotide species. And what we interestingly found is this: we found that the 25 to 35 nucleotide species is heavily enriched during the regeneration, during the initial parts of the regeneration, and they dramatically come down at the later stages, right? So the question is, do we see populations that are essentially enriched during the initial stages of regeneration? Do we see the small RNA populations that are enriched at the later stages? So in order to address that, we have created this line diagram here. What you see is a 25 to 30 nucleotide species can be broadly divided into 3 categories. The first category are that are highly enriched during the initial phases of regeneration, and they subsequently come down. The second phase is the ones that actually go up at 3 to 5 day time, and they come down at 7-day. And there is another population of 25 to 30 nucleotide species that subsequently go up as the regeneration progresses, suggesting that these 2 populations that you see here could potentially be playing a role in the tissue patterning and in the organogenesis. Similarly, we also found 18 to 25 nucleotide species that were heavily enriched in 3-day and 5-day regenerating animals here. And this could be essential either for the regeneration of the head, or in other words, these populations that you see here could be essential for the formation of the brain in the planarians, which needs to be dissected out. So we have identified tsRNAs that were enriched in planaria. We have identified that there are tsRNAs that are enriched during the head regeneration of the planaria, suggesting that they could be essential for brain formation. So the big question that we need to ask is, how are they made in the planarian populations, right? So the question comes here is, what makes these tsRNAs, these different species of tsRNA? As I said, we have these 5' tsRNAs, we have these ITUFs here. And we looked for angiogenin, which is traditionally shown to be essential for the breakdown of tRNA into tsRNA. And we haven't found angiogenin in the genome of planaria. So the next best bet we have is PV endonuclease. PV is an organette family of endonucleases that also cut the RNA and generate 25 to 30 nucleotide species in planaria. So what are these PVs? PVs are organette family of proteins, and they also generate 25 to 30 nucleotide small RNA species. And PV 1, 2, 3, there are three homologs of PV proteins, and all these 3 proteins are heavily enriched in the neoblast populations, and they were known to be essential for making the small RNA species, which are called as piRNAs, or they are also called as PV associated RNAs. The question we wanted to ask is, does this PV protein also cut the tRNA or not? And this has been well documented that these PV proteins are critical for the neoblast functions to happen. So if you knock down the PV, the neoblast doesn't proliferate and differentiate. As a result, you see a massive regeneration defect. This has already been shown, suggesting that PVs are critical for neoblast. And the question we are asking is, are they important for processing the tRNA? So the answer is very simple. Our work was being relaxed by the paper that recently came in 2020 in Genes and Development from Kloskan's lab. The Kim et al. Has taken each of this PV protein, and he has done pull down, and he annotated the small RNAs that are associated with these PV proteins. And if you look at this one criteria that he left here, which is called as unknown repeats, right? And no annotations. If you go back and look at what these no annotations are, it turns out that most of these no annotations that you see here are the 5' tsRNAs that are actually associated with the PV protein. And this is what you see here. You see here that both MEDV1, 2, and 3 are actually associated with the 5' tsRNA population, in addition to its association with the piRNAs that are derived from the mRNAs and the transposable elements. So what does this work show, this pull down show, is that the PV proteins, which are endoribonucleases, are indeed associated with these tsRNA populations, suggesting that the PV could be the protein that is essential for cutting the tRNA into a 5' tsRNA. So what, right? So going back to this slide, I said that when we take ESCs and differentiate them, we see two pools of tsRNAs here. One pool is angiogenin independent pool, other pool is an angiogenin dependent pool. And we don't know what is this angiogenin independent pool. So we went and asked, is PV expressed in the embryonic stem cells of mouse? And we found that there is one PV called PVL4, which is actually expressed or upregulated during the process of differentiation. So now we are doing this work, and we know that the angiogenin independent tsRNA pool actually could potentially be the PVL4 that is processing the tRNA into the tsRNA. This is something that we actually learn from the Planarian model. So going back, I just need another 3-4 minutes and I'll wrap up my talk. So I spoke about 5' tsRNAs that are derived from the PV proteins, right? Or potentially PV proteins are associated with 5' tsRNAs. And I have said that there are these ITUFs, which are intermediary tRNA populations derived right after the anticodon arm and extends into the T-C loop here. And we know that they show a strong enrichment for U, suggesting that these populations are either derived from dicer or organate. So what we have done is, we have went back and looked at dicers and organates in Planaria, and Planaria has one dicer and it has two organate proteins, right? And among the two organate proteins, what you see, EGO2, is the one that has been well characterized, and the EGO2 has been shown to be essential for microRNA pathway, and in conjunction with dicer, it actually regulates how the microRNA biogenesis is happening. We also found another EGO, which is very distinct from the organates that are present here, and they also have an endoribonuclease domain, which is called as catalytic triad, suggesting that they can potentially cut the RNA into small RNA species. So when we knock down dicer and organate 2, which is involved in microRNA pathway, we see a drastic regeneration defects. And we know both dicer and organate 2 here are essential for microRNA pathway. The question remains now is, what is this MED-EGO1 doing, right? So we went back and used single cell analysis, which is there from different groups that have done and deposited, and we have found that the EGO1, which is distinct from EGO2, has a very ubiquitous expression. It is expressed at a very low levels, but it indeed has a very ubiquitous expression. And when we knock down EGO2, we have seen a very mild regeneration defect. When I say mild, I mean that around 30% of the animals only showed a regeneration defect and others were fine. Okay, suggesting that the EGO2 is there, but its penetrance is low and it has a very low expression. So now in a knockdown animal, we ask the question, does it affect piRNAs and microRNAs? And what we found is, when you knock down EGO1, we don't see any disruption either in the piRNA levels or in microRNA levels. So the next question we asked is, does it perturb ITUFs and 5' tsRNAs? What we found is that the EGO1 knockdown actually perturbs or brings down the level of ITUFs, and they don't affect the 5' tsRNA, suggesting that the ITUFs that you see, which is 18 to 24 nucleotide population, is actually derived by the EGO2 protein, that EGO1 protein that goes and breaks the tRNA into tsRNA. So in summary, this is what it is. We identified a tsRNA populations. We identified 3 different tsRNA populations. One is 25 to 30, which is derived from the 5' arm, and other is I-tervs, and other is 5' tervs. And what we have shown here is this. By pull downs that came from Kloskan's lab, we have shown that the PVs can potentially cut the tRNA and can make tsRNAs. And we know that the PVs are expressed in the stem cell populations. So potentially we think that the 5' tsRNAs might regulate the stem cell functions, and they might be essential either for the maintenance of stem cells or for the differentiation of stem cells. And AGO1, in other hand, can cut this tRNA into I-tervs, right? And now these I-tervs can potentially behave like your microRNAs. And since we see there's a ubiquitous expression of AGO1, we think that the I-tervs might actually play a role in the later part of differentiation of stem cells into a specific cell types. And that needs to be investigated and proved. So in a nutshell, what did I summarize today? This is where I started my talk with, right? I started my talk with this. When you take a stem cell and give it a signal, the stem cells undergo state transitions. They undergo differentiation into progenitors and specific cell types. So what I have shown in last 1 hour is this. We have this ancient RNA molecule, which is called as a transfer RNA, which we all know carries amino acids with it. The other function that is emerging is that this tRNA molecule can break down into multiple pieces, and each piece can interestingly go and regulate translation in a ways that it can actually regulate how stem cells are differentiating into a specific progenitors. So that's what the summary of my talk today. And I need to thank 2 people here, especially, who has done amazing work in understanding tRNAs here. One is Shrikar. All the work, what you have seen right from the starting to the end, was actually done by Shrikar, and he has not just done the work, he has also put his brain to it. In fact, most of the Planarian part was driven by Shrikar and Vairavan. And Vairavan is a fantastic bioinformatician who has actually done a lot of bioinformatic work. He joined as a bioinformatician, but now he also does equally well the wet lab work. And both Shrikar and Vairavan are the key people who actually drove the whole tRNA project, tsRNA project in my lab. The initial part of the tsRNA work was done in collaboration with Dr. Ramanuj Dasgupta and Dr. Shrikala Raghavan. And the beautiful interactions between Shrikala and Ramanuj actually led to the evolution of this whole field in my lab. And been collaborating with Professor Alejandro and Kai, who now moved to China. And I have been closely working with Dr. Jochen Rink. And thanks to rest of the students, and thanks to the funding agency, which is Varna Jayanti, intramural funding from INSTEM, and thanks to Welcome DBT Intermediate Fellowship, which has been extremely supportive, mouse facility, NGS facility, and our microscopy facility. And thank you very much for your attention. I will be happy to take questions. Thank you.
Thank you, Das, for such an exciting talk to characterize this novel small non-coding RNAs that may have a role in stem cell differentiation. So we have received many questions, but I'll start with one of my own with the Planarian part. So as you have shown in the mouse ES cell differentiation, that the tsRNAs gets upregulated with differentiation. So do you see the same thing in the stem cell population of Planarians compared to the differentiated part?
Yeah, I mean, what we see here is this. I mean, we see that the tsRNAs are mostly associated with SMEDV3 than 2 and 1. So, I mean, in a way, so in a nutshell, we don't know whether this is upregulated when you take neoblast and differentiate.
Right. Okay.
And those are the clear. But by its association with these PV proteins, we think that it could be actually important to maintain the neoblast in its own state. right? Because we know that these PV proteins actually play an extremely important role in maintenance of the stem cells. So
Right.
we think that this tsRNAs might play a role there. But till we dissect the stem cell populations and look at tsRNAs, it's very difficult to predict currently, right? Yeah.
Right. Okay. So I'll take some of the questions from the audience. So it's a question by Shonchari Gorai. If tsRNA or the analog of it is found in the mammalian body, then can we use this for controlling the cancer stem cells?
Yeah, I mean, it's a good question. In fact, I haven't presented this. I mean, the tsRNAs are also heavily upregulated in cancer populations. So the tsRNAs actually could be a very interesting drug targets in order to control the cancer progression. Yes, answer is yes. Yeah.
Okay. So we'll take the next question by Prabhat Kumar Swen. Is there any specific cause to do methylation or demethylation during modification of enzyme by tsRNA? And how much time is required for this process?
So, I mean, I actually didn't get the first line here. Is there any specific cause to do methylation and demethylation? Okay, so let me answer this. The function of these modifications on tRNAs has not been very well studied. It's still a work in progress across to actually understand what is this modifications doing to the tRNA. But what is emerging is this: by looking at how this tRNA population can be broken down into small RNAs that can regulate translation. Now we feel one of the major functions of these modifications is to actually prevent or enhance this kind of breakdown happening, which is critical to regulate how the protein synthesis is happening, right? So now going back, how much time is required for this process? This is an extremely active process that happens inside a cell, right? As the tRNAs are being made and put out from the nucleus into the cytoplasm, these tRNAs come heavily modified. So now the important question that comes is, how is that such an active dynamic process has to be regulated? So we think that the regulation need not happen at the enzyme level, because you cannot inactivate an enzyme, because these enzymes also do many other things. For example, the DNMT2, which is also critical for modifying the DNA, right? You cannot inactivate the whole enzyme. The way you can control the enzyme activity is by modulating their cofactors, right? In one case, it's SAM, and methionine feeds into the SAM, and SAM is a methyl donor. So now you modulate the local concentration of the metabolite. You can control how the enzymes are acting. So though it's an actively dynamic process, which is methylation and demethylation, you can control the rate of methylation and demethylation by actively regulating the accessibility of the cofactors. So that's what we think is happening here. Yeah.
Okay. The next question is by Vinod Gupta. Are tsRNA can be derived other than tRNA?
Can derived other than, I mean, the name tsRNA suggests that they are derived from the tRNAs. They are tRNA derived small RNAs. So I mean, I assume if it is derived from any other molecule, I would rather call it by that name. So they're all derived from tRNAs. Yeah.
So next question is by Deepak Kumar Podia. Upregulation of tsRNA in turns reduces available amino acid tRNAs required for translation. I'm curious, how does this tsRNA pathways affect or bias global translational tool?
That's a great question, actually. Yeah, I mean, this is a question we get always from the reviewers back. So the question is simple: if tRNA is such a fundamental molecule that is critical for carrying amino acids, right, to add it to the growing peptide chain. Now I'm saying that this tRNA is being broken down. But if you now go back and compare how much of tsRNA is made and how much of tRNA is available, we only think that around 0.1% of a tRNA is actually being converted into a tsRNA. That makes the whole process more intriguing now, right? Now, you need to take a small fraction of the tRNA and convert it into a tsRNA. So now the regulation becomes much more complex. Now you cannot degrade the whole tRNA, you can only degrade a small fraction of it. So, so how is that happening? We still don't know that. So in a way, the answer to his question is, we are not actually depleting any tRNA. The tRNA is in so much excess. We are only taking a small proportion of the tRNA and we are converting it into a tsRNA. So now how is that regulated? We still don't know, and that's what we are trying to understand.
No, I think this is an excellent topic to work on, and how you have connected both the yes cell knowledge and the planarians. I mean, that's the beauty of using these non-model organisms to understand some of the fundamental biological questions. We have, I think we're running out of time, so thank you very much, Das, for doing this on a Sunday evening, and thanks to everyone for joining today. So we truly appreciate your support and for giving us this beautiful lecture today in the webinar series.
Thank you, Sonak. Thank you, Sonak, very much, and thanks to Dr. Reena for having me here, and thanks a lot. Right.
With that, I'll now request Professor Inurai Banerjee to give the vote of thanks.
A fascinating work, Dasaradhi. Simply fascinating work. I'll ask you a very basic question on behalf of students. Why Planaria?
Yeah, please. Why Planaria? So, why Planaria? I mean, I will go back and take some of the lines that were put by Alejandro. I mean, I really appreciate the way he gives his talks. So, one of his lectures, he said this, right? Our whole understanding of biology is driven by a handful of model organisms, right? There is so much there to learn and explore. And one example of that is Planarian model, right? I mean, this model, what it is telling us is that the plasticity and the flexibility that this model has to actually grow and make any tissues and organs is the amazing strength this model is offering to us. So there's a lot of fundamental biology that can be learned by looking at how a stem cell can make a specific tissue in the right place and the right time. I mean, I don't want to get into this controversial argument here, but one of the biggest drawbacks with, I would say drawbacks are a lacuna with stem cell biology field per se is you have a stem cell you can make into a specific tissue, right? But what you need to control in order to make it more therapeutically potential is to make it to a right amount, right place, in a right spatial situation, right? In other ways, you can make an organ in a tissue, in a plate, but if you take that organ and put it back into the body, can you actually connect it back to the existing tissue? That's one of the biggest challenges in regenerative medicine, right? What is this organism telling us is this: you can take this organism, you can cut the organism at any place. This organism has the capacity to regenerate the organ, right? It regenerates in an organ in such a way that it takes the new tissue that is formed and beautifully integrates with the existing tissue. So there's a lot of biology we can learn in terms of how tissue integration is happening, how the axes are set during regeneration, which is also very critical if you are actually taking a stem cell and making it as an organ in a plate, right? So that is what these organisms teach us, which we cannot learn otherwise, right? So that's what I say is important, why it is important to study Planaria. Right? Yeah. I think you are mute.
Now you're mute.
Yeah.
Yeah, it's fine now.
Okay. Okay. Yeah. So a mute animal like Planaria telling us so many things through the fascinating work like Dr. Dasaradhi here. So yes, I hope I'm unmuted. Yeah, as a biologist, especially as a zoologist, it is humbling to appreciate that when something works, how evolution preserves it. something works, something clicks, and it stays on and it is conserved. So I think that is the beauty of the fundamental studies that you have been doing. So thank you so much for sharing your work, and best wishes, because we expect many more interesting information to come out of the work that you are doing. So thank you so much, and thank you to all the audience who have been tuning in, and for all the great questions. And thank you, Dr. Dasaradhi, once more for giving such nicely rounded answers. This I must appreciate. So nicely you have taken all the questions and great answers.
Thank you.
So,
Thank you.
and thank you, Shonok, for
Yeah.
conducting it so nicely.
Yeah.
All right, so
I enjoyed my time here. Thank you very much for inviting me. It's a pleasure to be here. Thank you very much. Thank you.
And we hope you'll come back and share more of your
Sure.
ongoing stories as they unfold.
Definitely. And I also look forward to listen to you both actually at some point. Yeah. Thank you very much.
Thank you very much.
Yeah. Thank you.